Hello everyone, let's get started. Today we're going to be talking about the control of microbial growth. The first thing we need to ask ourselves is why would we want to control microbial growth in the world around us? The first and most obvious answer is in the prevention of disease. Of course, we want to be able to prevent infection due to microbes that might be on or in our bodies. But we also prevent disease by controlling the growth of microbes in our homes and, importantly, in our hospitals. A second reason that we might want to control the growth of microbes is to protect our food supply. We haven't talked much about the impact of pathogenic microbes on foodborne diseases. But it is an important area that impacts our health every day. We need to be able to protect our food at the source. In other words, we need to be able to control what's growing on plants and animal-based foods on the farm. We need to be able to control the growth of microbes in packaging facilities and at slaughterhouses. We even need to be able to control the growth of microbes in the places where our food is sold, both in restaurants and grocery stores. One other place where microbial growth is a very important consideration is anywhere that a clean space is required. And if you're not familiar with that terminology, when we're discussing industrial processes, we often need to have clean rooms. And those rooms are literally clean of any sort of particulate material. So if, for example, we were in a pharmaceutical manufacturing plant, the drugs are being put together and packaged in clean spaces. Places where computer components are being manufactured, those also have to be clean spaces. So there are lots of places in our everyday lives where controlling the growth of microbes is paramount. We do always want to remind ourselves, though, that most of the microbes that we encounter every day, both on and in our bodies and in the wider world, are not pathogens. Remember, most are not harmful to us. Now, there are some general ideas around this concept of controlling the growth of microbes that we need to understand. The first is that there's a big difference between trying to control the microbes in the world around us and trying to eliminate the microbes in the world around us. We will not be successful destroying and eliminating every single microbe. First of all, you wouldn't want to do that, right? As we know, the vast majority of the microbes that are on and in our bodies are actually important to us. They are critical to our normal, healthy functioning. We also know that there are all kinds of microbes out in the natural world that are critical for healthy ecosystems. Yes, there are times when we want to eliminate microbes from a particular environment. But most of the time, we're really just talking about controlling them, particularly controlling the amount of pathogenic microbes that we might be exposed to. Only the process of sterilization will completely kill and remove all of the microbes in a particular environment, including viruses. Only sterilization will accomplish that. We also need to understand that only certain forms of sterilization will destroy endospores. Remember, by their nature, endospores are extremely resistant to destruction. In order to destroy them, it's not enough to just apply high heat. You have to apply high heat and high pressure. And you have to apply those two things for a relatively long period of time to destroy spores. So It is possible to kill and remove microbes completely from surfaces. That's what sterilization is about. But oftentimes in our everyday lives, we don't need that level of control. There is some terminology that we need to be comfortable with because it turns out we can achieve different levels of control of microbial growth depending on how aggressive we are in our methods, particularly when it comes to chemical methods that we're using. I have 3 terms here that I think demonstrate this fact. The terms sanitizer, disinfectant, and antiseptic are terms that you may well have seen, especially as you walk down the cleaning aisle in the grocery store. Lots and lots of products are available for us to purchase, and they're labeled very differently depending on the product. A sanitizer, for example, is a product that will reduce the total number of microbes on surfaces, on inanimate objects and surfaces. So you might use a sanitizer, for example, to clean floors or to clean countertops. A disinfectant is a little more rigorous. A disinfectant is capable of eliminating the most common pathogens that we tend to encounter, again, on inanimate objects and surfaces. So you might want to purchase a disinfectant if you're cleaning a surface that you're concerned may end up contaminated with pathogenic microbes. Say, for example, cutting surfaces in your kitchen or surfaces in your bathroom. Now, finally, this term antiseptic, this is an important one because an antiseptic is a type of compound that you would apply to living tissue. We don't sanitize or disinfect living tissue. No, we use an antiseptic on living tissue to, again, eliminate common pathogens. So, for example, we would apply an antiseptic to a wound to help clean it out and ensure that we didn't leave pathogenic microbes in that wound that might lead to infection. The take-home message is that when you're buying products with the idea of using them to control microbial growth, you have to be aware of what that product is capable of. And that means you have to read the label because these terms mean different things. Now, even when sterilization might seem like a good option for certain environments or locations, we have to remember that sterilization is not always very practical. Number one, it can be expensive. The kinds of equipment and tools and chemicals and things that you need can be expensive. And number two, sterilization is harsh on materials. And sterilization can indeed destroy materials over time. So again, you have to understand what level of control you really need before you decide, for example, whether you ought to disinfect a surface or try to sterilize a surface. Now, there are lots and lots of different methods available to control microbial growth. We're going to talk about physical methods and chemical methods. Now, by physical methods, I mean heat, which is the most common physical method used, radiation, and filtration. Let's start with heat. Heat is a really interesting and really applicable method of controlling microbial growth because depending on how you use the heat, you can achieve sanitization, you can achieve disinfection, and you can achieve sterilization. So depending on what temperature you're able to hit and for how long, you'll get different levels of control. It turns out that all microbes have a range of temperatures in which they are capable of growing and at which they grow best. So when we use heat to control microbial growth, we're trying to hit temperatures above that range. Now, depending on which temperature we hit, and again, for how long, we can either reduce the growth of microbes, we can completely stop the growth of microbes, or we can kill the microbes. So again, we can tailor heating towards what end point we're trying to reach. Now, what exactly is high heat doing to a microbe, to destroy it, to kill it? There are two structures in a microbial cell, in fact, in any cell, that are most susceptible to heat. Number one is the membrane, and number two are proteins. If you're going to apply heat at a high enough temperature and for a long enough time, you will kill the cell. You will disrupt the membrane around the cell, and you will cause the proteins in the cell to denature or essentially fall apart. When it comes to the use of heat in controlling microbial growth, it turns out that the type of heat is very important. And by type, I mean moist heat versus dry heat. It turns out that moist heat is a better choice for controlling microbial growth, and that's because the moisture will help penetrate materials better than dry heat application at any given temperature. The conditions during heating are also very important, and what I mean by that is what exactly is in the material that we're trying to apply the heat to? What are the chemical conditions in that material? For example, acidic conditions during heating will increase the effectiveness of the heat. So imagine, for example, that you are trying to preserve foods for packaging. Maybe you are canning foods, and you have to apply heat to ensure that you have sterilized those foods. Acidic foods are essentially easier to heat sterilize. The heat is going to be more effective more readily when you are working with an acidic food than when you're working with a neutral pH food. So things like tomatoes and pickles and other very sour types of foods, because of their acidity, the heat that is applied to them is more effective. In other words, both the heat and the acid conditions are working against the microbes. Neutral pH foods, things like maybe corn or beans that you might package in a can, those are a little bit harder to apply the heat to. You have to work a little harder with the heat because you're only attacking the microbe with the heat. Interestingly, high amounts of sugar, high amounts of protein, and high amounts of fat in a food will also increase the effectiveness of the heat. We talked a little bit about sugar being a chemical condition that works against the growth of bacteria. If you recall, when a food has a very high sugar content, that sugar, that carbohydrate, tends to hold on to any of the water that might be available in that food and keep it away from the bacteria. And of course, that is a situation the bacteria can't survive in. It turns out that protein and fat are also detrimental to the growth of microbes. So any foods that are high in sugar, protein, or fat are a little bit easier to heat sterilize. Now, what about salt? We also talked about salt briefly, and recently we said that high levels of salt are detrimental to most bacteria. With the glaring exception of Staphylococcus, most bacteria can't tolerate high levels of salt. And by high, I mean anywhere from about 7.5% up to about 10% sodium chloride or beyond. It turns out that high amounts of salt, when they're used in combination with heat, can either increase the effectiveness of the heat or actually decrease the effectiveness of the heat. It all depends on the organism that you're trying to control, that you're trying to eliminate. Now, heat can be combined with pressure, and that will also increase the effectiveness in terms of being able to control the growth of microbes. And when we talk about heat and pressure together, we're talking about autoclaving. All autoclaving is a process that generates steam, heat, wet, moist heat, under pressure. And autoclaving is capable of sterilizing materials. Now, you have to achieve the correct combination of conditions for the correct amount of time in order to sterilize completely. You have to hit a temperature of 121 degrees centigrade at 15 psi, or pounds per square inch in terms of pressure, for at least 15 minutes. And if you achieve those conditions, you can sterilize the material. And remember, autoclaving not only can sterilize, but it can also destroy endospores. So it has a terrific practical application in medicine. Now, we don't have to limit ourselves to those three conditions: 121 degrees, 15 psi, 15 minutes. We can go beyond those. Higher temperature, higher pressure for a longer period of time if we need to. And sometimes, depending upon the thing that we're trying to sterilize, sometimes we need to do that. So if you look at the bottom of the slide, you can see some examples of wrapped versus unwrapped items that we're trying to sterilize in an autoclave. And you'll notice that all of these things have this white tape on them somewhere. Somewhere on the packages there's this tape. This is autoclave tape, and this tape is treated so that when the correct conditions are met, when you hit at least 121 centigrade and at least 15 psi for at least 15 minutes, you're going to get black bars appearing on this autoclave tape. You can see those bars over here on the left. So that tells you that these packages have already been autoclaved. You can imagine it's important to be able to know in a storeroom, for example, or an equipment room, which things have been autoclaved already and which things might be waiting to be autoclaved. And that's what the indicator tape will tell you. So whether you have unwrapped things or wrapped things will make a difference in your exact parameters for your autoclave. Things that are quite bulky and are wrapped up in a lot of soft materials, those things are just a little bit harder to sterilize. These are surgical packs. You may or may not have seen these where you work. Often we put together these packs depending on the surgical procedure that's going to be performed. There might be a pack, for example, for an appendectomy. And inside that pack there's going to be a tray with all the instruments that the surgeon is going to need to do that procedure, some sterile gauze, maybe some lap pads, the gown that the surgeon is going to need to wear. All of those things are packaged up in this blue material and then autoclaved. So when this package comes out of the autoclave, everything inside that blue wrapping remains sterile until that package is opened. Now, autoclaves come in all different shapes and sizes. I put this slide in because sometimes people are a little startled to see that there are autoclaves that are this big. You know, if you're working in a facility that just generates a whole lot of materials that need to be sterilized, you can encounter autoclaves of this size. You can also encounter much, much smaller autoclaves, again, depending upon where you work. Most doctor's offices, for example, have a small desktop autoclave in their facility to meet their needs. Very, very different from, say, an industrial-sized autoclave like this one. Recall that we also, in addition to autoclaving nice clean materials that we're preparing to use, we also autoclave waste materials. And of course, that's what you're looking at here. You can see there's a whole bunch of agar plates inside this autoclave bag. Perhaps this came out of a microbiology lab or a diagnostic lab. This entire bag will be sealed up and then placed in an autoclave in order to sterilize all of these materials that may otherwise be a source of contamination. And of course, down here we have a whole bunch of needles and gauze and sharps and things. We have to be able to autoclave waste in order to make it safe for disposal.